Okay, so thank you so much, everybody. We now have 75 people in the room, which is absolutely amazing. And I'm very happy to host our third Malaria Gen Journal Club. Thanks so much for John and for Pablo and Sri for helping us organize this. And today we have the pleasure of hosting Dr. Abebe Fola in our third journal club. Abebe is a postdoctoral research associate at Brown Institute at Brown University at Bailey's lab, and he's currently working on population genomics, drug and diagnostic resistance of malaria parasites. He obtained a master's degree in medical parasitology from Jimma University in Ethiopia. He has a PhD in malaria population genomics from Walter and Eliza Ho Institute from Melbourne, Australia. And he was a postdoctoral research for 2 years at Purdue University. So with that, very much welcome you, Dr. Abebe, and he's going to be talking to us about his latest paper that he published not long ago. So whenever you're ready, you're more than welcome to start your presentation. Thank you so much. Oh, and by the way, just one sec. There is one Q&A function at Zoom. So whenever people would like to ask questions and if you want to raise something, just please post on the Q&A session over there on the button. You will see the button on the bottom of your screen. Lovely. Are you okay? Baby, to start?
Yeah. Can you see my slide first before I start?
Oh, good.
Okay. Thank you, Christina and others for the invitation, and then thank you everybody, and then good morning, good evening, and good afternoon wherever you are. And as Christina said, my name is Abebe Fola, and I'm currently postdoctoral at Perila Brown University, and I will be presenting exploring malaria parasite drug and diagnostic resistance through genomics and then its implication for malaria control and elimination. And then as Christina mentioned, I will be focusing our recent publication. So my presentation outline, I will touch base a little bit on my journey, then malaria elimination challenge, especially drug and diagnostic resistance, and the importance of the large-scale genomics, and then the key molecular tools, especially we're using whole genome, amplicon, SNP barcode, and then so on, but I will be focusing on MIPS, which is we call molecular inversion probes. And I will also present what current MIP panel we have, and then mainly focusing on our genomic surveillance of drug and diagnostic resistance in Ethiopia, and the summary and the way forward. So to give a little bit my journey, so I was originally from Ethiopia, from south part of Ethiopia, from a small village where malaria is and was and is still endemic. And I did my BS in medical laboratory science in one of oldest university in Ethiopia, we call Gondar University. Then I did my master's in medical parasitology, where I started working on malaria. Then this is some of my highlights when I was doing my PhD in Australia. And then, so currently I'm a postdoc, and then my research area is mainly population genomics of malaria, drug diagnostic resistance, and then developing cheap multiplex molecular tools, especially amplicons and MIPS. And I also worked a lot in SNP, whole genome sequencing, MIP, and microsatellite analysis. So anyone currently working on this area, you are welcome and feel free to reach me out. So the key papers we will discussing today is the first paper, our paper in Ethiopia, where we found the parasite circulating in Ethiopia is carrying one of the key KLH mutation we call 622I, and then at the same time, the parasite is carrying HPR2 deletion. And after we published this paper within a month, another group in Eritrea, they confirmed that this parasite circulating in near the border area in Ethiopia where the Eritrea parasites were. They also showed using therapeutic efficacy study where in dietary positive cases are carrying this mutation. And then based on WHO criteria, this mutation is currently, it has been a candidate mutation, but now it's a validate mutation. And another group from Ethiopia, one of my colleagues from Sumtadese and his group, they found that another interesting finding that this the new mosquito, Anopheles stephensi, is also contributing the spread of this drug and diagnostic resistance in Horn of Africa. So it's really, I would say it's not good time for East Africa and Horn of Africa. So we have to be proactive and do a lot of surveillance, and we need a backup medicine for, I mean, it's really gonna be very bad if we lose our ACTs. So additionally, there is one paper which came out last week where you can see the prediction of the HPR2 deletions. So you can see, especially the Horn of Africa is like, I mean, the HPR2 deletion gonna be very high soon. So currently, Eritrea already changed non-HPR2 based RDTs. Ethiopia in the process of changing. So, and other countries are actively doing surveillance of HPR2 deletion. So to give a highlight what's happening currently in malaria, I think most of you guys are working on malaria, so this is gonna be not a new, but there was a significant reduction in malaria, but recently from the 2015, there is resurgence of malaria, and then the progress has stalled. And then what does it mean is that what the implication of the current landscape of malaria is that it's truly, we can say that expanding coverage of available tools alone unlikely eliminate malaria. So it requires new tools and novel strategy to achieve malaria elimination. So one of the major challenge, we do have a lot of challenges, but the main challenge is the mutation in KLH13 gene, which causes artemisinin resistance, is one of the major challenge. So artemisinin is one of the best therapy we have been using, and it's common in different combination in African setting, especially artemisinin lumefantrine quartem is the most common one. But the resistance has been reported in Southeast Asia in 2008. And you can see these are the KLH domains, and we have WHO candidate and validate markers. So each country, I will be presenting some of the key markers circulating in different parts of East Africa. But overall, the emergence of this KLH mutation is one of the main challenge. And then recently, the first report in KLH mutation, especially the validate mutation, was reported in Rwanda 2020, but the data is from 2000, I think 14 or 15. So when we look back to history of artemisinin resistance in Southeast Asia, I will be not discussing in detail because our today webinar is mainly on East Africa, but we have to learn what has happened in Southeast Asia. So it was detected in 2008. Then after that, the partner drug resistance happened, and finally the treatment and failure, and then the key mutation, especially spreading most of Southeast Asia, especially Great Mekong region. So now, after that, scientists have tried to assess, like in different parts of Africa, to see whether these key mutations are circulating in Africa, but until recent years, there are a lot of polymorphism, okay, but those polymorphisms in KLH are not either validated or candidate mutations. So as I said, the first report is the publication in 2020, where the parasite carrying one of the key mutation, what we call 561H in Rwanda, and which also associated with delayed parasite clearance. So in addition to the KLH mutation, because the webinar and then my topic gonna be mainly focusing on Eastern Horn of Africa, so you can see that we have more additional challenges, especially, so I borrowed this slide from the Dr. Fitz's presentation, where we have HPR2 deletion. As I said, we have a artemisinin resistance parasite circulating, and we have the urban mosquito Anopheles stephensi, which also currently shown that it's responsible for outbreak, and then the diagnostic drug-resistant parasites. And then more importantly, also in this area, Bivax and Parasparin are sympatric, so they coexist together. So this is additional challenge. So whenever we think about elimination of malaria in Horn of Africa, we have to think also Bivax, which contribute around 40+%. And due to different factors and the climate change, the highland malaria is also very common. And recently, this is also another big threat for malaria elimination, where one of the key mutation, which is 622I, which is gonna be the main focus of our presentation today. And there is increased prevalence of 622I, and it's co-occurrence with HPR2 deletion in Ethiopia, which is one of our study, and then the Arterian study, where it shows that the increased prevalence of this mutation in recent years. And then this parasite is also carrying HPR2 deletion. So when you see the state of this KLH mutation, you can see different mutations circulating. In Horn of Africa, you can see 622I, especially Eritrea and then Ethiopia. In Uganda, Rwanda, we have other key mutations like 675, 469, and 561H, which is common currently in Uganda, Rwanda, and Tanzania and the border areas. So what does it mean is East and Horn of Africa nation have revisited samples to search KLH mutation, and then it's very important to scale up genomic surveillance. And then also it's very important to assess artemisinin mutation spreading and its co-occurrence with HPR2 gene deletion. So to do this kind of large-scale genomic surveillance, here we have to focus on the genomics, because one thing I want to highlight is that malaria transmission dynamics it's quite complex, as you know, and then we will mainly focusing on genomics, but also we have to also focus on the vector part, the environment, the human genomics, and human parasite host environmental interaction is very important. But for this talk, we will mainly focusing on the genomics of malaria parasite. So the genomics of malaria parasite is very important to understand how the parasites respond to control intervention. And it's very important to detect early emergence spread of drug and diagnostic resistance. And it also facilitates the new and the novel tools to support malaria elimination strategy. So as I said, we have different tools to do genomic surveillance, or this where we call MMS, malaria molecular surveillance. So in our lab, we mainly using molecular inversion probes. So just I will give a little bit highlight. I'm not going in detail.
Abebe, can you hear us? I think we lost him. I think he's back.
Can you hear me?
Yes.
Okay. Can I continue?
Yes, please.
Okay. Okay. So just we're using this molecular inversion probes for the large scale genomic surveillance, but in simple term, these molecular inversion probes are there one type of the amplicon sequencing approach, but it's a little circular. But once you have the amplicons, you're gonna get the linear ones. So this has been optimized, and currently we implementing these molecular inversion probes in different countries in Africa and then beyond. So how it works is here you can see we do designing. So you have a ligation and extension arms where just think these are you like the primers. You have a UMI background, and then once you pull your MIPS, which is you can pull a thousand. Currently, we're pulling thousands of MIPS. Then once you have that, you add ligation and the polymerase. We call this is MIP capturing. And once you have MIP capturing, you do exonuclease digestion to remove circularized MIPS. Now, once you have that, just it's more of like adding more Illumina primers, the 4-1 barcode unique primers, which helps you to pull a lot of samples. So you can pull thousands of samples, thousands of probes, and then you do PCR cleaning, and then you do sequencing based on available technology you have in your lab, either HiSeq or MySeq or whatever. And then currently we have softwares to do downstream analysis, and feel free to look at these GitHub pages and reach some of the guys currently maintaining the GitHub here. And we do have a lot of MIPS, and I'm not going detailed, but anyone working beyond Plasmodium falciparum, we have MIPS for vivax, mosquito, and the human, and feel free to reach me out. But for this discussion, I will be focusing on, we call IBC or the genome-wide MIPS and the drug resistance to do drug resistance and to do background relatedness of the parasite. So here we can use MIPS for the different, either the drug resistant importation, long-range transmission, copy number, population structure, and related analysis. So the genomic surveillance of drug and diagnostic resistance, which is the main focus of this webinar, is that, so what we did in our study is that we took already genotype samples, which was published by Faleke and other colleagues in 3 regions of Ethiopia, Tigray, and Amhara, and Gambela. So we genotyped nearly 1,000 parasites from these 3 regions, and then we looked the key mutation, the 622I, and then we looked the association between the deletion and the drug resistance. So to do that, as I said, these are the sample sites and the number of samples shown in the bar plot here. So what we did was just we took these samples and we did genotyping with a genome-wide MIPS, which are around 2,000 MIP panel to look the parasite relatedness, parasite population structure. And then additional MIPS we call drug resistance MIPS, which is 800. So a total of 2,800 plus MIPS in a single tube. And the key drug resistant genes we looked at were CRT, DHFR, DHPS, KLH, MDR, and other background mutations. And after that, once we MIP and genome, Illumina sequencing, we took the FASTA-Q file, and then we have a in-house built panel, analysis panel or pipeline we call MIP tools and the MIP licorn. And then you can see after variant calling and filtering, we end up with the good samples and good loci for the downstream analysis. So the first result here is that the first finding from this study was the parasite circulating in Ethiopia in Syria region is a kind of low complexity infection with more than 70% of the cases carrying complexity of infection 1, which indicates low ongoing transmission, and then which is also one of the key thing that this HPR2 deletion and the clonal transmission is more common when you have low transmission, and that's what's happening in Ethiopia. And we did additional FW statistics to confirm this COI estimate, and we found the same finding. Majority of our samples have, FW is greater than 0.95, which is the monogenomic infection. And then we saw a little bit heterogeneity in the transmission, which are reflected by COI variation in 3 regions. And then the second finding was just this mutation has been reported a while ago in one region here, but we found that this mutation is spreading in 3 regions of Ethiopia. And then additional finding is that 80% of these parasites with the mutation, they're carrying HPR2 gene deletion. So imagine that the parasites, they're escaping the detection, and also they're escaping the treatment. In addition to this, 622I, one of the key mutations, because we have a marker which is more than 800. So we look at other key mutations, and then this the marker, that SP markers are really high in the country. And then so one thing I want to mention here is that in most of African countries, especially in East Africa, where CRT mutation is very low, but in Ethiopia and in Horn of Africa, you can see here that CRT mutation is very high because of the buybacks. Chloroquine is still in use in the country. Another finding here we found that is the mutation and the deletion, they are circulating, but they are not strongly associated. We found the mutation is more common in undeleted parasite than the deleted parasite. So the main reason could be that if the patient is carrying the mutation, so because of this, the current diagnosis and the treatment policy, if the patient is not, positive by the RDT, so this patient is not getting the ACT. So that's why in the case of deletion, less ACT pressure that could contribute. Moreover, maybe currently 6-7 mutation frequency is very low. So, and then it's also indicate that these two deletion and mutation they are independently or circulating, but the current finding in Eritrea shows that the opposite, where they are more circulating together. So the main reason Ethiopia is maybe the 622I prevalence is currently very low, why we seeing the co-occurrence is very low, but at least we found 80% of 622I mutation carrying HPR2 deletion. But one of the key finding we found in this study is that, which also supports our hypothesis, that if the patient is not getting ACT treatment, and this patient, because of the sign, the symptom, they also look alternative treatment. So they go to any private pharmacy and they get the chloroquine. So that contributes the high prevalence of CRT mutation. And we found that in our study that the delete parasite, either the double deletion or single delete parasites, carrying more CRT mutation than undelete parasite. That's another finding. Also, we found that, as I mentioned, in Southeast Asia, it's not only KLH mutation is contributing, but we have to also look at the partner drug resistance. So you can see here the upset plot showing that the 622I, if you look at this side, the 622I mutant parasites, they're carrying additional mutations that are indication for especially the lumefantrine decreased sensitivity, which is especially NFD, MDR, haplotype is one of the key. So the parasites with 622I mutant also they're carrying additional background mutation. And we look at, identity by descent analysis. So because of the time, I'm not going in detail, but so unlike the traditional method, like we used to look the frequency of the SNPs or like the what we call alleles, but here we're looking the shared allele. So the relatedness to ancestry is varies based on the recombination. So the more recent recombination and the more close recombination you get, more or longer IBD segment. So in our case, we found that evidence of clonal 622I transmission across site. The first thing was, if you look here, there is significant higher relatedness within mutant versus wild. The second thing, we look at this, the mutant parasite within the district. So this each color and each dot indicates, each dot indicates the parasite, each color indicates the district. So you can see, especially if you look at this district, then IBD, which is very high related parasites are there more, clustered within a district, which is really shows that the potential expansion and spreading of this, the mutant across the country, and then the clonal spreading at the district level. And we also found that the double delete parasites are, you can see these are it's very tiny, but you can see the significant difference. But the key message is that the double delete parasites are very highly related within, and you can see they are also more or less clonally transmit in the district level, which also one of the main challenge that these parasites are overtaking the, undelete parasites. So the key message is that, currently Ethiopia is also trying to use the new, non-HPR2 based RDTs, but what the finding indicates that these parasites are overtaking and undelete parasites because they are clonally transmitted at district level and they are spreading. And we looked at the population structure, and the population structure, we don't see any population structure per geography region, but what we found was the population structure based on the deletion status or either the mutant parasites. So you can see in this side, the mutant parasites are more clustering together. Either the single or double delete parasites are clustering together. So in summer, you can see that a significant spread of KLH mutation in multiple regions of Ethiopia, and also the neighboring arteria warrants large-scale genomic surveillance across Eastern Horn of Africa. And then the second key message from this finding is that the clonal transmission and spreading of the delete parasites at local scale causes a significant diagnostic challenge. So countries should consider non-HPR2 gene-based RDTs as alternative diagnostic tool. And the co-occurrence, which is the key message from these findings, that the co-occurrence of the 622I, the key mutation in the KLH with the deletion, which leads this strain potentially undetected by HPR2 based diagnostics, which warrants close monitoring of deletion and ACT resistance across Ethiopia and Horn of Africa. And also the finding also supports the utility of like nesting genomics data to National Malaria Survey. And then I will conclude with this slide. So as we all know that the traditional surveillance, it gives you a tip of iceberg where you can measure prevalence and incidence of infection, and it gives you a rough estimate, but it's less robust with, especially when in the low transmission area with declining transmission. And then it also does not inform the population genomic dynamics. That means like it's very hard to capture emergence and spread of the new variants. So the genomic surveillance, especially the large-scale genomic surveillance, is very important. It informs the genomic dynamics of underlying parasite population, how the parasite responds, intervention at genomic level, and how drug and diagnostic resistance is spread, the parasite connectivity, the efficacy of control intervention, the feasibility of control, and then you can see the corridors of parasite migration, and then it gives you more robust at genomic level. And then finally, I want to thank all the Bailey Lab members, especially Professor Jebeli and all other Bailey Lab members and other collaborators from UNC, the Ethiopian Public Health Institute, and especially I want to thank steady participants and their family, and then the funders, and then thank you. And then I'm happy to take if you have any question.
Thank you very much, Dr. Abebe. It's been a brilliant presentation, and it just highlighted the importance of genomic surveillance near real time. and then, you know, it's a ticking situation, isn't it? So let's see where science will take us to help public health decision making. So with that, I would like to also thank everybody. There was a time that I saw there was 104 participants in the room. I don't know, there could have been even more than that. So it's been a great attendance. So thank you very much, everybody, for coming over. And I will open the floor for questions. So you could either raise your hand or write your question on the Q&A session on the bottom of your screen. So then I'll alternate. Whoever raises their hands, and then we can go to the Q&A on the chat. So anybody with their hands up? Can we actually see when they?
I think we have one from Thomas Katiro.
Yes, so there are a couple of questions on the chat. So one of them is from Getu Hapte. He's saying, they are saying on the special event, I'd like to cooperate to work on anti-malarial drug discovery. So are you open for new collaborations, Abebe? How would that work for them if they wanted to work with you?
Yeah, always we are welcome. And also one thing I didn't mention is that, so this is our preliminary finding, and using this finding, we applied NIH grant, and we found recently new grant, R01, to expand this study to do more in vitro efficacy studies, and then to explore, because we did only in 3 regions of Ethiopia. Ethiopia is really a big country, so we want to expand this study. So there is a new research currently, and then we are always happy to collaborate and then talk and then work, not only even in Ethiopia, but all nearby and bordering countries. Thank you.
Thank you very much. There's Harvey, how has their hand up? If you want to unmute and ask your question, please.
There is hand from Hervé, I think.
Yeah. Can you hear me?
Yes.
Okay, thank you very much. Thank you very much, Christina. Thank you very much, Dr. Fola, for this nice presentation. I am Hervé Raoul Tazokon, PhD from Cameroon, PhD student. So it's true that I'm mainly interested in insecticide resistance in malaria vector, but also I have some main interest in parasite drug resistance and also diagnostic resistance. So yes, from your output or the main result that you have obtained so far, the allele 622I, based on the study that you have conducted, particularly in the Horn of Africa, at this stage, can you say that this mutation is a novel mutation, or it's spread from other locations from Africa, based on the fact that we did not screen this across Africa and temporally to see that maybe, for example, since 2000, for example, it was already present in some part of Africa and then it spread from that part to the Horn of Africa or something like that? Thank you.
Yeah, thank you for your question and really welcome to, I mean, truly speaking, we have to start collaborating like the mosquito and the malaria and human, as I said, just we have to work together. And for your question, I mean, so it's like, especially this mutation 622I has been reported in Ethiopia in 2004, 5. But since then, one thing I want to mention is that most of the time when you do this therapeutic efficacy and very few samples, it's very hard to capture the low frequent circulating parasites with this mutation, not only 622I, but we could have other mutations too. So you're gonna capture this kind of mutation when you do large scale genomic surveillance. And then whether this mutation is, I mean, whether it's really originating in Ethiopia or Eritrea or it's from other part of the country, I think it needs broader surveillance and doing, I mean, cross-country wide surveillance to see. But currently we can say that this mutation is not common in Southeast Asia, and it's more common in Horn of Africa, but more study needs to better understand the biology behind this mutation. Thank you.
That's it. Thank you. Thank you.
Thank you very much. So the next question is from Madi Safapur, and I'll apologize to everybody in case I mispronounce your name. I'm really, really apologize for that. So the question is, what are the advantages of MIPS compared to using Applicon or SNP panels?
Yeah, interesting question. Because yeah, the topic was not the MIP, but I have like a nice slide which summarizes how the MIP is. I mean, each method, truly speaking, I used to work through my PhD until today. I work on SNP barcode, microsatellite. I did a lot with microsatellite, whole genome sequencing, and then data analysis, amplicons, and now I'm working MIP and whole genome, all combined method. But truly speaking, each method has its own advantage and disadvantage. But the good side of MIP, if I want to talk MIP, so the first part is really highly multiplexable. So you can multiplex thousands of panel in a single tube. And you can also modify easily. Just if you have like, assume you found the new marker, and if you have like in all the panel, you didn't include that marker, just you can design new MIP panel and you can add, and then it does not affect your existing panel. So it's really highly multiplexable. And then truly speaking, it is the cheapest genotyping and sequencing method currently we have. And then I was comparing the MIP sequencing. If you multiplex the different sample and the panel, it might be more or less similar to the qPCR per sample. So it's cheap, highly multiplexable, and then it's really good to do, especially countrywide genomic surveillance, the big data genomic surveillance. So you can do thousands, and then we're currently doing like 10,000, especially in Tanzania, Uganda, Rwanda, and the Ethiopia. Currently we did 1,000, and currently we have additional 3,000 samples. So yeah, and then feel free, reach me out like to know more and to discuss more.
Brilliant. Thank you very much. Next question from Lemo Golasa. Hi, Lemo, by the way, hello. Well, he's saying, great talk. Thank you, Abebe. Is there any phenotypic genotypic association studies in connection with the 622Y SNP identified in Ethiopia?
Thank you, Dr. Lemu. By the way, Dr. Lemu is my master supervisor who introduced me how to start working on malaria. So I'm glad to answer his question and to discuss with him. Yeah, I think that's where currently just we showed this mutation is spreading, and it is co-occurring with 622I. That's the main, I mean, the deletion is occurring with 622I. That's the main finding of the Nature Micro paper. But as I said, the R01 grant we got recently is to look the genotype-phenotype association. So just the work has already started, and then we're gonna have a meeting soon in Ethiopia to discuss, and then I mean, the main aim of the grant is especially doing in-country parasite short-term culturing and to do more.
Thank you very much. Next question is, thanks for the nice presentation. Do you by chance check the origin of resistance in Ethiopia, whether it's imported or de novo?
Good question. Truly speaking, I mean, it's not imported like we used to say the mutation originates Southeast Asia and is imported to, I mean, Africa is not like that, but the current scenario is that either this parasite is circulating within Ethiopia or like Horn of Africa. So the origin is in Horn of Africa, but it needs like it's either it's originating in Ethiopia or Eritrea or Sudan or Djibouti, whatever. So cross-country nearby Horn Africa and East Africa analysis is very important, but I would say that it's de novo in Horn of Africa, but the origin, I have no answer for the origin. So it's most likely in Horn of Africa, most likely either Ethiopia or Eritrea or Sudan.
Thank you. The next one is from Vicky Simpson. Excellent presentation, Abebe. Thank you. She says, what are your thoughts on using multiple techniques for genomic surveillance versus everyone using the same technique or platform?
Thank you, Vic. This is a really high-level question, but so always I love, especially not only genomic surveillance. If we look all the malaria community, we don't have even like a uniform standard parasite quantification qPCR method. So each laboratory, we use different technique, different method, but truly speaking, it's good to have, I mean, one standardized method, but it's really very hard to say, okay, I'm like more comfortable with MIP, and it's very hard to say everybody should use MIP. But the key thing is that either you use amplicon or whole genome based on your, I mean, bioinformatics skill, your capacity, your sequencing, whatever. The key thing is that what you're trying to address or what trying to answer the question, the in the goal is very important. So either we use X or Y or Z method. If we're looking like, okay, looking the KLH mutation emerging and the spread of KLH mutation, we can do that one either. I mean, digital droplet PCR or amplicon sequencing or whole genome sequencing or SNP or MIP. But if you ask me personally, having one standard method is always good, but it's very hard to implement in all laboratory in the same standardized method.
Thank you very much. Next question is well, they're saying it's nice presentation. Did you compare samples collected from individuals with and without anti-malaria drug exposure?
That's a really good question. So we didn't look in our data explicitly whether those patients received treatment or not. So that's where the follow-up study currently, we have the therapeutic efficacy data, and then the follow-up and find either this parasite, I mean, the mutation is common in the DICE3 or after DICE3 positive samples and things like that. But if you look back to Eritrean study where they found this one. If you look back, they looked the parasite is common in DICE3 positive cases. So the same thing we're planning to do in Ethiopia currently, but it has been confirmed in Eritrea that this parasite is common in DICE3. Yeah, if you look this paper, they did the in vivo parasite assay and also therapeutic efficacy where the parasite is common in DICE3 positive cases. So, but we haven't looked that one in Ethiopia, where currently we're planning to start the same study with more in-depth analysis answering biology behind the deletion and the mutation in Ethiopia.
Thank you. The next one from the chat is from Takeli Teklu. So they're saying, thank you for your nice presentation. What do you recommend to develop country as large genome survey is expensive?
Yeah, also, yeah, it's good to, Dr. Takeli Teklu is also one of my mentor during my undergrad study. So it's really, so yeah, I mean, truly speaking, with the technology, the platform, we have like Oxford Nanopore sequencing, where we call a SITK sequencer. And then currently, I myself and our lab is currently trying to adopt MIP technology, MIP sequencing to Nanopore. So I would say sequencing is, I would say, is not gonna be expensive in near future with the support from Africa CDC, other collaborators. I think, I mean, countries like Ethiopia and other neighboring country, we can do large scale genomic sequencing. And then in near future, I mean, genomic surveillance gonna be not expensive. I would say we might also start even doing in using our smartphones and then like that. So currently it seems expensive, but it's gonna be not expensive in near future. And I have hope, and then even everybody gonna do, even kids can do sequencing in near future too. So thank you.
Thank you. Next one from Ryan Kaguruzi. They're saying, nice presentation, Dr. Abebe. I have seen you were successfully able to run the HRP2/3 assay to determine diagnostic resistance. What protocol did you use? We have tried the multiplex John Parr and Jen Moss protocols, but the HRP2 in particular fails to amplify. The HumBB is also problematic for us. HRP3 and LDH work fine. He says, thanks, Brian from Uganda.
Really, that's a really good question. So one thing I want to mention is that, so one of the collaborators, the main lab, Dr. Jonathan Parr and IDLE group, they are currently WHO certified laboratory for HPR2 deletion assay. So if anyone currently using their assay, and if they have any issue, we are happy to discuss in detail. But one thing I was discussing here is that we have also a MIP panel where you can do the deletion. So just you genotype, you do sequencing, and you look at the deletion. So we're using that the Dr. Jonathan Parr and other standardized multiplex panel qPCR assay, plus we have a MIP panel to confirm the deletion. So, I mean, feel free to reach me out and then we can discuss in detail.
Next question from the chat is from LMIU Tauto. Interesting to hear from the nice presenter and how malaria may challenge high and low land. So do you think, is there difference in gene and species difference? Sorry, I think they want to ask, so do you think there's any difference between gene and species in both locations?
Is he saying in high and lowland malaria case?
Yeah, I think so.
Okay. I mean, truly speaking, in this study, we haven't explicitly looked at the high and the lowland malaria case, but because of like the different temperature and then different mosquito species and then most likely the distribution of, if we look the two main, yeah, we used to say Vivax and Parasparine, but Ovale and malaria is common in Ethiopia too, but I would expect the distribution of Vivax and Parasparine, I'm talking the terms of proportion, might be different in highland and lowland, but I haven't explicitly looked at what the proportion of Parasparine in highland versus lowland or buybacks on but truly speaking, the proportion, I guess it might be different in high and lowland, but I haven't looked at.
Great, thank you. And I just saw that Jonathan Parra, thanks Jonathan for answering. Brian, he just posted an answer to you at the Q&A that he is very helpful. He's very happy to help and troubleshoot with you. So there's a question whether certificates will be provided, and the answer is no, we don't provide certificates. This is just a webinar for discussing Journal Club for now. Doesn't mean that we can't look at it in the future, but for now we are not positioned to provide any certificates. So the next question is for Abera, it's from Abera Kumalo. Thank you, Dr. Abebe, for your nice presentation. Now you are suffering a lot due to drug resistance, especially in southern Ethiopia. Do you think this may be associated with K13 mutation spread? What is your recommendation to prevent this challenge?
Oh, I'm getting a lot of questions from my colleagues. So Abara Kumalo is one of my colleagues, and then so just truly speaking, the south part of Ethiopia, especially Dr. Lemmu and then Dr. Par and others are here. So in new study, we have to expand the south part of Ethiopia. I also got the call from my family, but I'm from the south, so there are a lot of, kind of malaria cases. So it could be, we never know, it could be drug resistance. It's very hard to say it's because of drug resistance, but I suggest anyone currently working in Ethiopia, and we can also chat and then discuss in detail, but we have to expand, do an in-depth analysis and then a genomic surveillance, especially the south part of Ethiopia. And then, the new project we currently starting soon, personally, I will also recommend to get more sample from south part of Ethiopia. And thank you, Abera, for the, information. And I have also information that there are a lot of cases. It's kind of a resurgence of malaria in south part of Ethiopia. So it could be different factor, but it's very hard to say due to drug resistance. But it's very important to expand genomic surveillance and other study, especially south part of Ethiopia.
And there's a follow-up question and comment from Lemu. Thinking beyond the box, he says, wouldn't it be possible to look at other regions of the parasite genome that may have associated with ACT's delayed parasite clearance in Ethiopia, Africa, as there might be de novo mutations?
Yeah, thank you, Dr. Lemu. That's a really interesting question. So I try to touch base, especially, it's not only KLH mutation, it's just we have to look closely the partner drug and then like other genes. So and in our panel, we have more than 14 genes. So just I present very key ones, but yes, it's very important to look how the 622I or other KLH mutation with other, more of what Dr. Lemu was saying, looking other background mutation and then looking the haplotypes. So yeah, that's really very important. And I'm happy to chat with you offline and discuss more and to get your recommendation. And then thank you very much.
Thank you. I'll just read the last two questions and then we'll have to stop for now. But the penultimate one would be from Sabine Milong. They are saying, thank you very much for your nice presentation. I am a PhD student from Cameroon, working also on a PFK13 gene. I found also novel mutations on the couch propeller domain from my Cameroonian isolates using only forward strand for segment sequencing. Your presentation is very useful and inspiring for my future research work to achieve my PhD thesis. Can I contact you by email for more orientation, mostly on sequence analysis?
Sure, more than happy. And then the novel mutation, non-synonymous mutation, there are always polymorphisms there, but that does not mean it's validated or candidate. But truly speaking, feel free to email. And then Christina, I think you guys, can I share my email, or this attendees they have my email? So
of course you can. Yeah, absolutely.
yeah. So yeah, I can put in the chat my email, but anyone feel free to contact me.
And I'm sure if people Google your name, they will very easily find you as well.
Of course, yeah.
Right. So the very last question, which I think will be it would finish today in a really good tone, which is from Fisea Wada. Dear Abe, is there a possibility to see a malaria-free world in my lifetime?
That was, if you look some of my, during my PhD, and my kind of highlights were like seeing malaria-free world in my lifetime, less hope, and at least less days, and then malaria, most country free from malaria, I would say. We will see that one. Thank
Brilliant.
you.
Thank you so much. Abebe, it's been excellent. And I'm sure we still have 71 people around, lots of comments saying how wonderful your presentation was. So thank you so much again for your time, for sharing your knowledge here with us. It's been a real pleasure to host you and to have you. And we will be advertising soon the next Malaria Gen Journal Club for the end of